this I thought was the mark scheme. Ribose nucleotides instead of deoxyribose. Uracil instead of thymine, we've said that. Single-stranded, although it can fold into three-dimensional structures. Oh, shorter than DNA. Okay. And there are three kinds. Three? We've probably only come across two, really. mRNA. I don't think the third one is particularly on your syllabus anymore. So mRNA, tRNA are the two you want. There's also something called ribosomal RNA. Okay, is the third one. Okay, which you don't need to know a great deal about, but the clue's in the name. There is a type of RNA that makes up the ribosome. Okay, and you literally don't need to know any more than that, folks. But in this question, we're going to go with, well, there's three kinds of mRNA, tRNA, ribosomal RNA. The other thing, this is a lot less stable and less protected than that. And obviously its role is going to be to go into the nucleus, pick up the copy from the DNA, and then leave the nucleus. And the important point about that is that the DNA never leaves the nucleus. So not only is the DNA really stable and really well protected, it all stays in the nucleus and doesn't move from one place to the other. And so that means less of the cell is occupied by DNA than you might think, and therefore it's harder to, let's say, hit that DNA with radiation. You know, most of the time we are exposed to some radiation, background radiation, and even more so when we take a flight. But a lot of the time it just passes through our cells, and as a consequence of the DNA being coiled and compact, the radiation can pass through our cells without ever striking the DNA there. So it's there protected in the nucleus. Similarly from chemical reaction, but there we have it. So I did want to talk a little bit about tRNA. Okay, so let's look at this drawing of it. Now, it has to be said that this is a simplification of tRNA. So first of all, we're saying, well, tRNA has about 80 nitrogenous bases. Okay, it's worth remembering that. It's worth remembering, of course, with it being RNA, it does use U as well. And it's worth saying that at this end, we will attach an amino acid. Okay, and at this end, we have something called an anticodon formed up of 3 bases, and that will attach to always mRNA. Okay, so the mRNA provides a code to the amino acid. Now, the truth of this is that that structure is a 2-dimensional structure. The reality is, it would actually, these clover leaves would actually fold over, and it ends up as a sort of an R structure. Okay, and so on one end here you'd have the connection with the mRNA, and on this side you would connect with an amino acid. Okay, now it turns out that if you think about what these tRNA is doing, there needs to be 20 different tRNA molecules. Okay, it needs to be 20 different. So where does the difference come? Well, slightly different sequences here, and perhaps here and here, means it folds up slightly differently. And so, like everything in nature, the shape there, the shape of this tRNA is going to determine which amino acid, which enzyme, because an enzyme is going to join the amino acid here to the tRNA, and then the tRNA is going to join to the mRNA here. Okay, and so the function of the tRNA then is to bind with a specific amino acid. How is that specificity achieved? It's about getting the right shape of the tRNA that will fit into the enzyme that catalyzes this reaction to join a specific amino acid to the tRNA. So at that point, that shape leads to 20 different, 20, and is mainly around here, 20 slightly different shaped tRNA molecules with a general shape. You need to focus. That's probably a bit more than you need to know. But the general shape you need to be aware of is it's clover-leaved. It's at this point here, it's sort of single-stranded. At this point here, it's double-stranded. At this point here, it's double-stranded. Then it's single-stranded. It's single-stranded. Then it's double-stranded. So you can see what we said before about it's rather a special case. But this whole cloverleaf, it's called in 2D, we can call this a cloverleaf structure. You don't really need to know on your syllabus the fact that that will further fold over to make a 3D structure. And you don't need to know that, but just the point you do need to know is there's 20 different ones, and they're specific to 20 different amino acids, folks. And that is achieved by, as nearly everything in nature is sometimes, I feel, is achieved by shapes. Okay, the shape gets things done, folks. Okay, the way shapes interact, if I were to break it down. Okay, so that's a discussion of the tRNA. Obviously, got the amino acid on the end there. If you could, if you had an amino acid here, it would react with a neighboring amino acid there. That's why you'd be thinking, if it didn't fold up further, you think, well, these are a bit far apart, aren't they? How are they ever gonna react? Well, the amino acids on the end of the R, they can get quite close. This amino acid here can get close to this amino acid here, and they've got to get close together to form a condensation reaction there. And a peptide bond, which is the ultimate purpose of all this, is to join amino acids together into a protein with a peptide bond via condensation. So in a way, you need that extra. Well, how is that bringing two amino acids together? They're miles apart, so look at all these clover. They'll fold, folks. They'll fold further. Okay, it's just a little bit above the syllabus, but sometimes that helps you understand better what's going on. Okay, so ribosomal RNA, I think this is beyond what we need to know as well, but I think it's worth, what you will need to know is ribosomes in eukaryotes have two subunits, small and large, and are assembled in the nucleolus of the nucleus. So that's a part that we do need to know. Okay, so the ribosomes, actually, you'll find them in the cytoplasm or on the endoplasmic reticulum, rough endoplasmic reticulum. You'll find the ribosomes there. They're all made in the nucleolus, folks, so don't forget that. And there's a lovely picture of a ribosome. And much of that ribosome is actually made of ribosomal RNA. Okay, so it's almost like the ribosomal RNA and the tRNA are almost structural, like a protein. Okay, and this diagram, and I won't worry about it too much, it's all of these RNAs, tRNA, mRNA, rRNA, are actually made in the nucleus and then exported into the cytoplasm. Okay, and so the RNA forms the ribosome there. Okay.